All right, so welcome to the online lecture. Like I mentioned before, we're going to use this material for an in-class assessment, so please pay attention, take notes. Like I said, if we do have the in-class assessment, you can only use your notes that you've done or your slides. So we're going to start here, which we talked about, the cell being like a factory. So every organelle I mentioned is going to be somewhat of a factory, and I'll give you some examples of how that relates. So again, the cytoskeleton, this is like the building walls or the building structure. So if you think of any type of factory, the frame around the building is going to do the same thing the cytoskeleton will do. So let's skip forward because we covered this in class. The cytoskeleton has about 3 components. So this image here is a really nice image showing all 3 of them, and we're going to cover them. The largest one, so if you're making a list, the largest one is the microtubules. So micro usually means small, but in this particular case, it's the largest one. Think of a tube. Think of something pretty big, like a hollow tube. A microtubule is going to be the largest, so it's 25 nanometers. But it's a long cylinder. It's a long hollow cylinder. And you're going to typically find the microtubules in the core of the cell. So right in the middle of the cell, near the nucleus, is where you would find the microtubules. They are a little bit different because they have this characteristic called dynamic instability. So if you've heard of the term dynamic, that usually means like a lot of movement or a lot of going on. Instability is where something is unstable. So you basically can add or subtract to the either end of the microtubule to give it length. So because this structure is involved with movement, when we're adding to one side or when we're subtracting from the other side, we call that dynamic instability. So dynamic because it's a lot going on, and it's unstable or instable because we're adding things, we're subtracting things, we're adding things, we're subtracting things. So all of this is going to cause that term dynamic instability to happen. With the intermediate filaments, they are number 2. So you have microtubules at the top, and then intermediate filaments are the second biggest. And these are smaller at 10 nanometer. Think of these as being twisted like a rope. So if you think of like a Twizzler or anything like that, it's going to be twisted and rope-like, and they're going to be spread out across the cell. So they're not just at the core where the nucleus is, but it's spread out across the entire cell. With actin filaments, we also call these microfilaments. So I know that's a little confusing because the first one is microtubules, but remember a tube is kind of big. So think of a microtubule as being the big part and the microfilament as being smaller. So these are the thinnest part of the cytoskeleton, and they form these spiral, thin, long strands. So you can find these on the edges of a cell. So remember, the core of our cell, you're going to have microtubules, but the edge of the cell, you have microfilaments. Microfilaments also have another name called actin, A-C-T-I-N. It's a very, very common name in cells. We use that to make sure we're talking about the same number of cells because that number shouldn't change. So this is an important aspect of biology. Again, this is just showing the picture. You have some things that are going to be close to the nucleus, which is blue, and then you have some aspects of cytoskeleton that's going to be on the outside. This is also an image from your book showing the cytoskeletal parts from largest, 25 nanometers, to medium to smallest with actin. So again, this is going to help you. Remember microtubules, if you're making a quick list, microtubules are hollow and they're big, and they have these dynamic instabilities, so sides that grow and sides that get smaller. Intermediate filaments are in the middle and they're like twisted. And then finally, actin filaments are on the edge of the cell, and they're the smallest and the thinnest. All right, so we checked off the building of our factory. So let's go into some other things you may have heard of too. So the first one is flagella. So we talked about flagella in class. With flagella, you're usually going to see long single flagella, but sometimes you can see flagella in pairs. Flagella are going to move in a whip-like or wave-like fashion. So think of a wave going like up and down. Flagella are going to move that way because it's useful for movement. So flagella is specific to movement. If you were to cut off the flagella of bacteria, it would not move at all. That's because flagella is important for movement. So this is also something specific to prokaryotes. So when we do our Venn diagrams, this is something you can include in your prokaryote side. We do have one exception, and that exception is sperm. So in eukaryotes, we have sperm, or males have sperm, and sperm, as you've seen before and as we talked about, have one flagella behind them. So normally if you see flagella, it's a sign of a bacteria, but if you see the flagella, it could be with a eukaryote. So maybe a multiple choice question says, you see an organism that has a flagella. What group could this belong to? Prokaryote, eukaryote, or not enough information to determine? So what would that be? You identify a substance that has the flagella. Is it prokaryote, eukaryote, or not enough information? The answer would be not enough information because you can't tell if it's a prokaryote like bacteria or if it happened to be a sperm. So that's one type of sample question with this. The other type of structure that we're talking about is cilia. So cilia are really short, and think of them as like hair-like projections. So if you were to think of something like a kiwi that has just hair all over the surface, this is what cilia is like. So instead of moving in like a wave or a whip-like fashion, they kind of move in an oar fashion. So oar is kind of left to right, left to right. So they are going to be really useful for adhesion. So like we talked about, if you were to throw something on the wall and it has a lot of little small sticky cilia on it, it's going to have a higher chance of sticking than if it had nothing on it. Again, this is a picture. This shows the sperm with the flagella, and this shows paramecium with cilia. All right, hopefully you're doing okay with the online lecture. If anything was confusing, this is a great time to pause and rewind and try to get caught up to speed. All right, so the nucleus and the endomembrane system. So now this portion, I'm going to talk to you about some of the actual organelles that you're going to find inside the cell. So we're basically going to start here in the nucleus. We're going to work our way through the ER and then also talk about the Golgi apparatus. So before I get there, I want to tell you what the endomembrane system is, because that could be a little confusing. The endomembrane system is basically a connection or a network of membranes that start off at the nucleus, and it moves through the ER, it moves through the Golgi, it moves through the lysosomes, the vacuoles, and the plasma membrane. So we're starting off at the nucleus, and if you think about the movement of, let's say, proteins through the cell, this allows it to get from the nucleus to the ER, to the Golgi, to vesicles, to finally the plasma membrane. So some things are directly connected, like we'll talk about the nucleus and the ER, and some things are not. So between the ER and the Golgi, they are not connected. So again, what is the endomembrane system? It's just a connection or a system of communication from the nucleus all the way to the outside part of the cell or the plasma membrane. So let's dive into this nucleus, because I've been saying that a lot. I'm going to give you some information on that. So the nuclear envelope is basically this double membrane, meaning we have two layers, right? So if you see here in this picture, you see it's a layer on the inside, a layer on the outside. It is a double membrane layer that is around the nucleus, and it's going to keep all of the nuclear components on the inside. So the outer side of the nuclear envelope is directly connected to the ER. So that's a great question. What organelle is directly connected to the nucleus? And it is the ER is directly connected. Nuclear pores allow passageway to go through. So all of the nucleus has little holes in it. Because we're going to make stuff inside the nucleus, we need to eventually get it out. So that's why we have pores. And this portion here is not really super critical. So portions of the nucleus are not in the endomembrane system. That part is not super critical for you to know. So once you jump inside the nucleus, you're going to find some items. The first thing is a chromosome. So a chromosome, we used to see in chromosomes looking like X. I can't draw on this, but an X. So chromosomes inside the nucleus are going to be an X. But a chromosome is made up of DNA and proteins in a tightly compacted state. Whenever we're looking at the DNA and proteins itself, when it's not compacted, we call that chromatin. So remember, chromosomes are tight and they're very structured, while chromatin is loose. So think of chromatin as being loose, and chromosomes as being tight. So chromatin loose, chromosomes tight. So try to remember that, because then you might see it a little bit later. All right, nuclear matrix. This is basically just the area that's inside of the nucleus. Think of it like a scaffold. It's just an area that everything can kind of sit in place in. So it's basically like the internal structure of the nucleus. This is really critical. So ribosome assembly, where do we make a ribosome at? That occurs in the nucleolus. So we make ribosomes in the nucleolus. I'll tell you what that is in a second, but it's basically the nucleus of the nucleus. It's the core of the nucleus, okay? So again, really critical if you have a question that says, you remove the nucleolus of an organism, what organelle would be decreased? Would it be the ER? Would it be the ribosome? Would it be the cytosol? Would it be the plasma membrane? It's going to be the ribosome, because remember, the ribosome assembly occurs in the nucleolus. All right, so our nucleus, if we're thinking of a factory, we can assume our nucleus is like the boss or the CEO. They have all of the genetic material. They make all the decisions on what needs to happen throughout the cell. Again, thinking about your Venn diagrams, we start talking about some of these organelles, especially with plants and animals. They're going to have some similarities, so just kind of keep that in mind. All right, like I mentioned, a ribosome, I mentioned this a little bit earlier, but a ribosome are going to be identified as little tiny dots on a picture of a cell. But more importantly, they are really critical for making proteins. So ribosomes are the site of protein synthesis, or the site of protein creation. Scientifically, we call that term translation. So if you ever get what is another term for protein synthesis, it would be translation. If you get translation means what as it relates to biology, it would be protein synthesis. So all cells, no matter what, prokaryotic, eukaryotic, bacterial, animals, plants, whatever, they are going to have ribosomes. So this is something all cells have. So again, our ribosomes, think of them as the employees of the factory. They do all the work. They're the one that gets things done. So again, now that we've left the nucleus, right, we're in the nucleus. We made our ribosomes in the nucleolus, which is like the core. The ribosomes leave the nucleus through a nuclear pore, and they directly go to the ER, because the ER is directly connected. So the ER is a system of like flat, think of them as like a stack of pancakes, kind of a stack of flat fluid-filled tubes, or we call them cisternae, but tubes. And what happens is once the ribosomes leave the nucleus, they go to the ER. Now there are two types of ER. You might be able to see it in this picture. There's the ER that has the ribosomes on them, and the ER that does not have ribosomes on them. So the ER with ribosomes on it, we call it rough ER. So RER, rough ER. It's rough because it has something on it. That something is the ribosome. So it's going to be studded or covered or saturated with ribosomes. The rough ER is directly connected to the nucleus. So again, that multiple choice question, what is directly connected to the nucleus? The best answer is not ER, but the best answer is rough ER. not smooth ER. So just keep that in mind. And then one of the major functions that the rough ER has is protein assembly. So please don't get protein assembly mixed up with protein production. So remember we did our examples in class, the sheet of paper, that was the primary structure of proteins. The ribosome is what does that. The ribosome gives it the code ATCC, AT type of thing. But when we're talking about protein assembly, like taking the piece of paper and folding it and bending it and all of that, that is actually done in the rough ER. So that's a really big differentiation to make between where the assembly happens and then where the production happens. So although the rough ER is responsible for folding of the protein, it obviously needs to work really closely with the ribosome because that's where the protein is made. So usually the ribosome makes the protein, and as soon as it makes it, the rough ER works by folding it and putting it in the proper place. So again, it helps with protein folding. Once it is done with that protein folding, it can then send that protein to where it needs to go, and we'll talk about that in just a second. The alternative one is the smooth ER. That's here without anything on it. It's called smooth because it's free of ribosomes, so it has no ribosomes on it. So it does have some major functions. One is metabolism. So metabolism is what we're going to talk about a lot, but that's how you make energy for the cell. So imagine if you had work to do. Think of a table that's crowded. It's going to be really hard to do your work. But if you took a table and you spread it out and there was nothing on the table, it would be very easy for you to actually do the work. So think of that as surface area. The rough ER is crowded with ribosomes, but the smooth ER is smooth because there's no ribosomes, and this is going to allow more work to be done. So that surface area you can use for reactions. The storage of calcium occurs here. So let's say you get a question that says you have a decrease in calcium after some type of disease. What organelle is probably responsible? It's the smooth ER. And then finally, lipid synthesis and modification. So again, you have a disease or one of those scenarios I give you, you look at a cell under the microscope, it has no phospholipids. What organelle was probably the cause of this? It's probably the smooth ER because that's responsible for photosynthesis development or steroid development. So again, the ER is like the assembly line. So we get the product, and as we're putting it together like you would in a factory, that's where the ER comes into place. The Golgi apparatus, it has almost the same exact structure as the ER. However, the big thing is it is separate. So if you're looking at a picture, you can tell the ER from the Golgi because the ER is directly connected to the nucleus, while the Golgi is separate. The Golgi is going to be apart. It's going to have some space between the rough ER and the rough ER and the Golgi. There's going to be a little bit of space there. So the Golgi has a really big job in helping to sort, process, and secrete protein. So you see in this picture here, all of the work of the folding and creation of the proteins happen here. Once they're done, they get into these little circles, these little kind of shells. We call that a vesicle. So it's like a balloon, basically, that's carrying the protein around. Once it leaves the ER, it then directly connects to the Golgi. And in the Golgi, this is where it kind of packages the protein to where it needs to go. So if you made a protein in your brain and it needs to get to your eye, then it might add some type of special tag that says, when you leave the cell, go to the eye. Or when you leave the cell, don't stop anywhere else, go directly to where I need you to go. So this is going to be kind of like adding a label to a package. Let's say you had to make a package and you added a label and you tell people where it needs to go in the world. This is what the Golgi does. It helps by sorting and processing and then secreting it eventually. So the Golgi is like shipping and receiving. So it's going to receive what's on the assembly line, and then it's going to add a label to it and a tag to it and send it to where it needs to go. The lysosome, like I mentioned before, this is going to only be in animal cells, and it's important for breaking down something that's not supposed to be there. So it's usually full of acid or some type of acidic compound or enzyme that will break things down, dissolve things down, so that the host in the body can get rid of junk and then, be functional. So lysosome will be like waste management, right? They're the things that go around and kind of pick up the trash and get things situated. So that's essentially the same thing, right? Gets rid of trash or materials. Vacuoles we'll talk about a bit briefly. So with vacuoles, both animal and plants have vacuoles, but we just call them different names. So with our animal cells, we call vacuoles storage vacuoles. So they don't have really a fun name or anything like that. They're just storage vesicles, and they're used to store material. So a great example is an adipocyte. So if you haven't heard of an adipocyte is useful for storing fat. So when you lose weight, it's not like you lose fat cells. You just lose the amount of fat that's held within this vacuole, right? So you're just losing either a little bit of fat, the vacuole is getting smaller, or the vacuole is getting bigger. That's basically how it works. With our plant cells, we have a central vacuole. So remember, animal cells is just storage. We just call it storage. With plant cells, they have a vacuole, but this one in particular is called a central vacuole. So a central vacuole does not store material, it stores water. So like I was saying, once you feed your plant, if you give your plant water, what's going to happen is that that central vacuole is going to plump up. It's like a balloon. You blow up a balloon, it's going to get really big. And once it gets really big, it kind of exerts or puts pressure on the cell membrane and makes the plant sit up straight. We call that turgor pressure, but it makes the plant sit up straight. Now, if you forget to water your plant and there's no water, the central vacuole is going to start to lose that water, and then your plant will start to slump over. So that's the purpose of a central vacuole, but because plants need water so much, it's so important for them to have like a storage place of water. Another type of vacuole that protists use, so you might remember that from when we talked about it a while ago, is a contractile vacuole. So this one is different because a storage and a central vacuole are used for storage, used for kind of holding or storing something. But a contractile vacuole is useful for pushing water out from movement. So think of a balloon. If you were to fill a balloon up with, let's say, air, and then you squeeze out the air really fast, it would actually propel the balloon forward, right? Like you blow up a balloon, you let it go, it moves forward. The same exact thing happens for water, but once you basically squeeze the vacuole, then the water leaves, and that movement of water leaving, the movement of you squeezing it, is going to move the vacuole a little bit forward. So again, this is just a picture showing the ones that we covered so far. Peroxisomes are simple. They are useful for breaking down H2O2, which is hydrogen peroxide. So hydrogen peroxide is very bad, but we have peroxisomes in our cells, which contains an enzyme that breaks down H2O2 into water and oxygen, which, we are fine with. So I think at this point we are pretty set. I'm going to actually stop here and not give you too much more, and we will cover the rest in class. So thank you for viewing this, and again, make sure you take your notes. Make sure you're kind of recognizing the color of this pointer. Maybe that'll be a question for you, but regardless, thank you again for doing that, and looking forward to seeing you in class.